in that what we have seen is what are various phases. G1 phase, then the S phase, metaphase, G2 phase, metaphase, etc. We have seen various important aspects of these phases. Then we have seen that how various checkpoints are there, and cyclin dependent kinases, they are playing very important role as a checkpoints, as a correctors, and as a regulator of the uh, one after another process in the cell cycle. We have also seen that until and unless it is sure, it is confirmed that everything is correct, it will not progress into the next one. And because of this, the next daughter cells, they are particularly having no errors in their genetic makeup. However, if this system fails, we have seen if it is a problem, then it can result into any kind of problem, or it can be even cancers, and we have seen various kind of situations in that. Then we have seen that whether there are some inhibitors are available for that. Yes, there are various inhibitors are there. Just like promoters, there are inhibitors which will inhibit the cell cycle, and these were CIPs, that is CDK interactin protein, and kinase inhibitory proteins. These are two important proteins are there, and these are families actually of these CIK families, KIP families, and other one is INK4A and ARF families are there, which are responsible for that. And in that we have seen that various proteins are there. P21, I told you these are the proteins and they play important role, and in this we have also seen that there are some synthetic inhibitors, or that what we can call as a drug, which are under study, or very recently some of these are marketed, hence they may not appear in books. So these are various agents which are used for various kind of cancers. So this is our various clibs, that is palbociclib, then ribociclib, then abemaciclib, then these are various agents. Then hormone receptor positive and HER negative breast cancer, they are used. Okay, so these are various agent ways of working in this area, so that scientists are finding that this can be better, this can be safer one. Then we'll not go for this transcriptional regulatory network, how the transcription is regulated, and there are many factors are there into that. The basic studies are going on in the this fungi, that is Saccharomyces cerevisiae. In that it is there, and how this is happening, it is there. There are variety of transcription factors are there, which are important. They are periodically expressed, and these transcription factors, as the name indicates, we shall be again studying the transcriptor factors later in more somewhere more in details. There are many transcriptor factors are there, and in that they are important in the what we can say very specific transcriptions. You know about transcription, what it is, that is from DNA to RNA. What is the importance of it? That RNA is formed, then that will be responsible for protein synthesis, and accordingly the protein will be formed. So they are very important. This whole process is called, we call it as a gene expression. Gene is expressed, or gene is rather process for either replication or transcription. So these expressions takes place. The expression profile of these transcription factors are driven by the again another transcriptor factor. They peak in the prior phase. So suppose in next phase, suppose in G2 phase whatsoever are required, or in synthesis phase whatsoever things are required, then in G1 phase this transcription factor will be triggering the synthesis of that particular proteins or enzymes which are responsible or which are important. Okay. So these are such activities which are taking place. Many experiments are going on, and particularly how these cyclins can be modulated in various kind of activities. The studies are going on, so we'll not go into this detail. Whenever we'll encounter, we'll check it. Then next is again studies are going on about DNA replication and the DNA replication origin activities, how this DNA replication and the origin activities are going on. Now next thing important is these checkpoints. We discuss about it, that these are very very important. They are actually making the corrections. They are checking everything whether it is error free and try to remove the error. We have seen that we have got a mechanism to repair the DNAs, so they try to make the repair of it, and they are there to monitor the and regulate the progress of the cell cycle. That is, if everything is fine in G1, it is confirmed, then it will go to the S phase. If everything is fine, everything is correct, perfect, then S phase will go into G2 phase. And then if everything is correct, then it will go for the mitosis. So this is how the checkpoints are checking at every what we can say period or every cycle of cell cycle. And if required, they can go for the DNA damage. If they find that there is any DNA damage, why DNA should be damaged? There can be many things happening. If you remember replication, we have seen earlier, we have seen that there are two-way lagging phase and leading phase. In lagging phase, we have seen that there are the pieces of DNA are coming and they are joined by ligase enzyme. Now what can happen is this, during while joining, there may chances that one or other this nucleotide has changed, and it can result into whole big change in the RNA or DNA, the subsequent DNA or RNA. So this is how these DNAs are there. If they are damaged because of one or other reason, they should be corrected or they are corrected. Cell will not proceed to the next phase until checkpoint requirements have been met. Checkpoint typically consists of a network of regulatory proteins, and we know what regulatory proteins are there. They are the cyclin and cyclin dependent kinases. They are playing important role. There are several checkpoints that ensure that damaged or incomplete DNA is not passed to the daughter cell. So we have seen where these are: G1 to S, G2 to M. They are the checkpoints, and lastly is the metaphase or mitotic checkpoint. So these three important checkpoints are there, and these are ensuring that the correct daughter cells are formed. Okay. The G1 to S transition is rate limiting step in cell cycle. Rate limiting step you might have studied somewhere in kinetics or etc. Rate limiting step is the step which is taking the longest time, or on which the actual rate of reaction depends, if you have studied kinetics. So rate limiting step, that means which is a very important step, that is taking maximum time, and that is counting for the whole period that takes maximum one. So it is a rate limiting step, and it is also therefore known as a restriction point when G1 to S is there. And this is where the cell checks whether it has enough raw material to fully replicate its DNA, whether sufficient number of nucleotide bases are there, whether the enzymes like DNA synthesis or polymerases are there, whether chromatin is there, and everything is checked, and then it will go for that. Unhealthy or malnourished cell will get stuck at this checkpoint. The cell will not proceed beyond this. Okay. Then the G2M checkpoint, where the cell ensures that it has got enough cytoplasm and the phospholipids for two daughter cell, then only, because in synthesis we have already seen that replication has done. Now what it comes in G2 is before mitosis, which that means the sufficient of cytoplasm and the organelles are to be formed. So enough cytoplasm is there, enough phospholipids are there to form the cell membrane, and it checks to see that it is right time to replicate. Okay. There are some situation where many cells need all to replicate simultaneously. That is, growing embryo should have systematic cell distribution until it reaches to the mid blastula transition. So this is again done by control by G2 to M1. Okay. So these are simultaneously many cells are replicating, and some are going into differentiation if we see during infant growth. The metaphase checkpoint is a minor checkpoint because we have seen again in cell cycle that it has got a little time. The time wise it is just 10% or 9% rather we say. So this is just 9% and everything has been checked during G1 phase after synthesis and at the end of G2. So it is a minor checkpoint that one cell is in a metaphase. It is committed to undergo mitosis. That means it cannot be reverted back. Okay. Uh, Although it is minor or it is taking lesser time, but it is also important. In what sense is it ensures that the spindle has formed and all chromosomes are aligned at the spindle equator before anaphase begins. So these are important things which are taking place. While there are many these three main checkpoints, all cells do not pass through each of these. In some cases some are there, some may not be there. And many types of cancer are caused by mutation that allow the cells to speed through the various checkpoints. Just like people, some police is given the signal to stop and the people with motorcycle run away, उसके आजू बाजू से निकल के भाग जाते हैं वगैरह. So although there are checkpoint, but this some of this during because of the mutation or because of the structures of protein which are there, so the cells speed through the various checkpoints and even skip all these checkpoints, and so they go from S to M phase to S phase almost consecutively. So almost G1, G2 is not there. So synthesis of that is to replication takes place. Then this mitosis takes place. New cells are formed. So whatsoever requirement was there, that is G1, G2 was all preparatory phases. These are actually preparatory phases. So no preparation is there, and therefore the cell will continuously grow into numbers, but the size may not be grown and functions may not be there. So because of this, the cells, they have lost their checkpoints. Any DNA mutation that may not have been occurred, they are disregarded. So any changes in the DNA are there, they are not checked, they are not verified, and then it passes as continuously to the daughter cells. Okay. So this is one reason why cancer cells have tendency to exponentially accrue mutations. So therefore the cancer cells are continuously having mutations one after another because there is no checkpoint, and therefore the daughter cells are getting different from the parent cell. Apart from that, many fully differentiated cell cycle no longer replicate, so they leave the cell cycle and they go into G0 till death. We have seen this example of neuron earlier, and thus removing the need of cellular checkpoints. So in that there are no checkpoint because it is not going for any kind of cell division thereafter. The alternative model of the cell cycle response to the DNA damage has also been proposed. It is known as a post-replication checkpoint. So even after replication also, the scientists are believing that it can be checked, and post-replication the checking is taking place, and still DNA can be corrected. So this checkpoint regulation that plays important role in the organism's development in sexual reproduction. When egg fertilization occurs, the sperm binds to egg, it releases the signaling factor that notify the egg has been fertilized. Among that other thing, that this induces new fertilized oocyte to return from its previously dormant G0 phase to G1 phase, and then goes for the mitotic replication and other activities. We have seen about P53 as a protein. It plays an important role in triggering the control mechanism both in G1 phase, that is G1 to S, and G2 to mitosis phase as a checkpoint. In addition to this P53, the checkpoint regulators are being heavily researched for their roles in the cancer growth and the proliferations. So these are important things. This is how the techniques are there. This is known as a fluorescence imaging of the cell cycle. So these are various dyes, and how these are checked, how various cells and cell division see. You can see the this fluorescence, and it is from one cell to two daughter cells are formed. So this type of cell division and structures are there. It is making visualization. and this gives idea about cell division. So how these two daughter cells are forming and how they are separating. Okay, so these are various activities which are there. Then the important why we are studying again, since they are important in cell divisions and cell multiplication, and if anything goes wrong, we know that it is resulting into pathological conditions. So why we are studying that it plays very important role in tumor formation. So whenever this cell cycle is not properly regulated, or if it lost its control, then it may lead to tumor formation. And when some genes like cell inhibitors, we have seen this RB, that is retinoblastoma cells, P53, etc., when they mutate, they may cause the cell to multiply uncontrollably. So if it is not controlled, then it will be growing, and it will be growing beyond the size which has been limited, or that for which is given for that particular organ. So our body has got anatomically very fixed structure, and every organ has given their size, and accordingly it should not go beyond the size. But then if multiply uncontrollably, then at some places wherever it is happening, then it will grow, that particular portion, that particular part of the organ will go, and that is we call it as a tumor. So it will form the tumor. The duration of cell cycle in tumor cell is equal to, or it is longer than that of normal cell cycle, but the proportion of cell which are having active cell division, the in tumor is much higher than the normal tissue. Okay, cell cycle duration may remain same. If it is taking maybe half day, then in all there will be also taking half days. Okay, so the duration is same, or maybe it can be longer than the normal cell cycle, but the proportion of cell which are in active cell division, suppose as I told you, why cell division is required, we have seen it as a part of growth and repair. Now growth and repair means what? Example, in GIT, it is having always a wear and tear. So because of wear and tear, these tissues are continuously lost, and since these are continuously lost, then the problem happens is this is the this tissue has to be, some of the cells has to be reproduced, so that this tissue is in a continuation. Okay, so these cells are in what? Cell division mode. Then this will go into cell division, so that wherever these cells are damaged, these new cells are be taking place of this. But if this rate is more than what the cell damage is there, so this higher rate will be causing for a tumor formation. So there is a net increase in the cell number, as the number of cells that die by apoptosis or senescence that remain same. So cells which were dying, as I told you, are ruptured or damaged because of wear and tear or any reason. Now these cells having a fixed number, or same, maybe suppose any tissue is there, and there the cells are dying, 10 cells are dying normally, so 10 cells should be formed. But instead of that, if the 100 cells are forming, then it will form a tumor. The cells which are actively undergoing cell cycle, they are targeted in a cancer therapy, as a DNA is relatively exposed during cell division, and hence they are susceptible to the damage by drugs or radiation. So this fact is made use of in cancer treatment, that is process that is known as a debulking. So what is this debulking is? It is a reduction of as much as the bulk volume of the tumor without the intention to complete eradication. So it is usually by surgical removal. So if tumor is there, most of the part is removed from that surgically. And if it is done by using a very cooling or freezing, that is this has got so many methods. And if the surgical debulking of tumor is known as a cytoreduction, surgically removal of this, they are called as a cytoreduction. So significant mass of tumor is removed, which is pushing the remaining tumor cells from G0 to G1 phase, and this debulking procedure will kill these cells which are newly entered into cell cycle. Okay, so after that, radiation or chemotherapy has to be done to kill the cell. So this is normally removal of the tumor is there. The fastest cycling mammalian cell in the culture, crypt cell in the intestinal epithelium, have cycle time as short as 9 to 10 hours. Okay, because intestinal cells, they are continuously in a wear and tear, because food is coming, they are churning. Food may have some abrasive material, and therefore the cell membrane may be damaged. It is an internal mucosa. Even skin can get, skin is that much tough, still can be there. So these epithelial cells, they have cycling time now 9 to 10 hours. Stem cell in the resting mouse skin may have cycle time of more than 200 times. Okay, that means it takes 200 times to have a daughter cells. So these are due to the varying length of G1, okay, or that will stop in a G1, that will be remaining, just like as we have seen, you might have seen in microorganisms, that they go in a what we can say, in some cases you call them as cyst formation if it is protozoal etc. are there. Okay, that means they remain in a hibernated form. They will not expose themselves, or they will not show any activity, and they will be in a resting period. So in general, cells are most which are most radiosynthesis, they are late in metaphase and G2 phases, and the most resistant in the late S phase. Okay, so during S phase, radiation will not able to do anything to them. In cancer studies, we'll see the radiation etc. and they will be most radiosensitive in the M phase and late M phase and G2 phase. The cells with longer cell cycle time and a significantly longer G1 phase, there is a second peak of the resistance in the late G1 phase. The pattern of resistance and sensitivity correlate with the level of sulfhydryl compounds, that is known as SH groups. Okay, they are natural substances that protect cell from the radiation damage and tend to be at their highest level in S, and they are lowest during the mitosis. So these are various aspects related to that. Homologous recombination is an accurate process for repairing DNA. There are two processes for that, homologous and non-homologous end joining. Homologous is an accurate process, and this homologous is nearly absent in G1 phase, and it is most active in the S phase, that is synthesis phase, or where replication takes place, and it declines in a G2 to M phase, that is during G2 or mitosis it is minimum. The non-homologous end joining is a less accurate and more mutagenic process. Means, suppose if you remember tissue repair, somewhere if you have last year in pathology you have studied, okay, so in tissue repair you might have seen that there are two ways: the normal tissue is formed, or the fibrous tissue is formed, or we call it as scar tissue. Okay, if it is a very fast turnaround mechanism, then usually it goes for a scar tissue formation, and scar tissue has not a parenchymal tissue, it does not have functions. In liver also, at many places you might have seen this similar thing over here is. So if it is a homologous recombination, it may be somewhat slower, but it is an accurate process. While non-homologous end joining, it is a less accurate, but it is more mutagenic process, and but it is active throughout the cell cycle. While HR is absent, almost absent in G1 phase, and only more active in GS phase, and again it goes down to in next pages. So suppose in some cells, if this non-homologous end joining that is activated during G1 phase, because homologous is not active over there, so in that case what will happen is it will result into formation, or there are chances, that is we do not say that always it will do, but the chances of having a mutagenesis is more. And we know that what will happen if there is a mutagenesis. The mutagenesis will result into any kind of change in the DNA or genetic sequences, and that may lead to the problems. So here we finish the topic, that is cell cycle and various things which are associated with the cell cycle. I am very sure if you go through it in neatly, it will help you in during your anti-cancer drugs, and even these processes indirectly or in similar way, if you study this bacterial cell division, then whatsoever antibacterial drugs are there, they are acting on these processes, So I suggest you that you please go through these processes well, and then the next topic again is we'll take it separately, because today I thought that if we can try it, but if we go for again, it will remain half complete. Instead, we'll go for this topic in our next class. So see that you are going through this properly, read it and make the notes. uh, I have one suggestion also, that can you please start making questions also.